Hello. Today I'm going over cell respiration in detail lecture. So we had previously covered photosynthesis, and the process of cell respiration is kind of the opposite of photosynthesis. So in photosynthesis, the plant takes energy from the sun, and it takes CO2 from the atmosphere, and it's going to make a glucose or a food molecule you can think about like a sugar. That food can then be used by the plant or something that eats the plant. It also makes oxygen. In cell respiration, we're going to see kind of the reverse going on. So let's begin. This is how we get energy to move and do things. So when I say we, I'm talking about living organisms. All go through cell respiration in order to get energy from our foods. So glycolysis begins in the cytoplasm, where one glucose molecule is broken down into two molecules of pyruvate through a series of enzyme-controlled reactions. So we're starting with glucose. Think of this as the food that you eat, okay? It gets broken down, and eventually you get this glucose molecule, this sugar. It's happening in the cytoplasm. The cytoplasm is just the kind of like jelly liquid inside of the cell. So we're not in any kind of organelle yet. This process does not require oxygen and is therefore considered anaerobic. During glycolysis, a small amount of energy is captured in the form of two ATP molecules and two NADH molecules. So we see that the glucose gets broken apart using ATP, and it makes these two smaller molecules, 3 carbons from the 6 carbons that were there. Ultimately, what we're trying to do in cell respiration is make ATP. ATP is going to be that molecule that our bodies use in order to do work, move muscles, etc. Let's see. We notice that it takes 2 ATP for this process. They're used up, but we do get 4 ATP that come out of it. So there's actually a net increase of 2 ATP, but we need a lot more than that. So this really is about making these two 3 carbon molecules at the end. Glycolysis is the first stage of cell respiration and provides the starting materials for the next step when oxygen is available. So these will move on to the next phase. Down here, this is a different cycle and pathway, but you see the pyruvates that we started with now is up here at the top. The Krebs cycle, also called the citric acid cycle, takes place in the mitochondria after pyruvate is converted into acetyl-CoA. So we've got our pyruvate here. We kick off a carbon. This is going to form CO2. And here we're actually dropping off, we're picking up electrons in this process. So it leaves me with a 2-carbon acetyl-CoA. The acetyl-CoA combines with a 4-carbon molecule to form citrate, beginning a series of chemical reactions. So you see this one is going to mix, they're going to combine together. So 1, 2, 3, 4, 5, 6 over here. As the cycle continues, carbon atoms are released as carbon dioxide, which is a waste product. You see that here, here, and here. Just giving that's the carbon dioxide that we breathe out in our breath. That's where that carbon dioxide comes from, is this waste product of this process. The energy released is stored in the high energy molecules NADH, FADH2, and a small amount of ATP. So at each step of the way here with these NADHs and FADH2, we're putting energy into those molecules. We're adding energy through an electron. And we can produce, again, a small amount of ATP, but not enough. We made a little bit up here and we made a little bit there, but more to come. Okay. The Krebs cycle repeats once for each acetyl-CoA molecule produced from glucose, meaning it runs twice for every glucose molecule. And this is the 2x. Since the glucose is 6 carbons, and it's going to make 2 of these 3 carbon pyruvates, for each glucose, it goes through this twice. Each turn regenerates the 4 carbon molecule needed to start the cycle again. So that's why you can follow this along, and you'll get another 4 carbon to meet up with the acetyl-CoA. The NADH and FADH2 molecules carry high energy electrons to the electron transport chain, where most ATP is generated. So that's where we're ultimately going towards, is electron transport chain, where the ATP that we need is going to be made at the end. Together with glycolysis, the Krebs cycle plays a vital role in converting the chemical energy stored in glucose into usable energy for the cell. All right, so again, we took energy from food, the glucose, to use in the cell. In photosynthesis, it was reversed. You're taking energy from the sun and putting it into a food molecule like glucose. The electron transport chain takes place in the inner membrane of the mitochondria and is the final stage of cellular respiration. So now we're in that bean-shaped organelle mitochondria. For these last two, we have been the electron transport chain and the Krebs cycle. Glycolysis took place outside of the mitochondria. So now we're in that organelle. High energy electrons carried by NADH and FADH2 are passed through a series of protein complexes embedded in the membrane. And we see this little area here kind of blown up. And here's my NADH and FADH2. They're going to give off these high energy electrons that now start to move through the cell membrane. As the electrons move from one complex to the next, they release energy. So these electrons are releasing energy, releasing energy, releasing energy. Well, that energy can do work. This energy is used to pump hydrogen ions from the mitochondrial matrix into the inner membrane space. So we use the energy from the electrons to move these H+ ions up into this area here. And you see we're getting a big collection of those hydrogen ions, a whole bunch of them. The movement of hydrogen ions creates a concentration gradient, storing potential energy across the inner mitochondrial membrane. So concentration gradient just means there's a high concentration and a low concentration somewhere. In this case, our high concentration's up here, and our low concentration is down here. This is important because naturally the high concentration will want to move to the lower concentration, and that can do work. The hydrogen ions then flow back into the matrix through a protein called ATP synthase. That's here. So as they go through, they're going to cause this protein to do some work, put some things together. ATP synthase uses the energy from this flow to combine ATP, or ADP, and a phosphate into ATP. That's our ATP. Our goal was to make this ATP. This process, known as chemoosmosis, produces the majority of the ATP made during cellular respiration. At the end of the electron transport chain, so we're over here, oxygen acts as the final electron acceptor by combining with electrons and hydrogen ions to form water. So our hydrogens come through here. Something has to be done with those, and what they're going to do is they're going to link up with this oxygen, some electrons, the hydrogen, and eventually they're going to form water. So you have to have oxygen. We breathe oxygen in for this purpose here, to be this final electron acceptor in this chain. That's why you have to have oxygen. As a result, ATP production would decrease dramatically, leaving the cell with only a small amount of ATP made during glycolysis. The electron transport chain is the most efficient stage of cellular respiration, producing about 26 to 28 ATP molecules from a single glucose molecule. So again, taking the energy from our food and through a series of processes, getting this molecule ATP, which now is going to be able to let your body do work, let your body move things around and go through chemical processes. Okay, there is more to this, but this is the level I want to approach this at. You'll have to probably review this a couple of times before it starts sinking in, because there's a lot to take in there, but it does move very logically. So take a look at it more than once, give yourself time to start to absorb it. Okay, if you have any questions, let me know. Thanks.